Gated ligand ion channels for acetylcholine and its behaviors by Ethan Yee and Matthew Zhao. Video number 303.
We will be talking about the neurotransmitter acetylcholine and its receptor, a ligand-gated ion channel. Acetylcholine signaling is used for thinking and muscle contraction.
Gated ligand ion channels use ligands that are hydrophilic as they bind to the binding sites that are on the membranes outside of the cell. The receptor proteins are embedded in the cell membrane. Acetylcholine is hydrophilic, and its receptor is embedded in the cell membrane too. These receptors' responses are used for changing the concentration gradient of the ions, and it stops once it reaches dynamic equilibrium or the ligand detaches from the binding site. An advantage of these receptor proteins are that they are rapid and able to respond quickly to stimuli, but the disadvantage of them is that their response is short-lived. These receptor proteins work by when they are inactive, the gates are closed, and the channels do not let the ions flow through. However, when a ligand binds to the binding site, the channel opens and ions are able to flow through until there is dynamic equilibrium or the ligand unbinds, causing the channel to close again.
Acetylcholine is the signal used in this pathway. It is an agonist for nicotinic acetylcholine receptors, abbreviated as NACHRS. NACHRS are open when activated and allow positive ions such as Na+, K+, and Ca2+ to pass through. Acetylcholine, abbreviated ACH, or other agonists of the receptor form bonds at the binding site on the outside of the cell membrane. When an agonist binds to it, the receptor activates and changes conformation, opening a channel that positive ions can pass through. The movement of ions through the channel is driven by an electrochemical gradient. The response is terminated when one of two things happen. The enzyme acetylcholinesterase, ACHE, separates ACH from its receptor with hydrolysis, returning the channel to its original closed structure and stopping the flow of ions. Alternatively, the flow of ions stops by itself when the electrochemical gradient is exhausted and the inside and outside of the cell have equal charge. The flow of ions into the target cell has many effects, including altering the state of nerve cells through depolarization or causing the contraction of muscle cells. Its effective depolarization is a key for neuron communication and thus thinking.
Acetylcholine is an agonist because once it binds to the binding site of the protein, it initiates the cell response. But some ligands act as antagonists, which binds to the binding site but doesn't elicit a response in the cell. Some ligands are exogenous, meaning that they are from outside of the body. But acetylcholine is endogenous because our bodies produce it and use it as a neurotransmitter.
Agonists affect the signaling pathway by opening the channel to let more ions into the cell, and because there is more of that, it will mimic the effects of acetylcholine, such as muscle movements. Antagonists affect the signaling pathway by binding to the binding site of the receptor without activating it, and this creates competition for the acetylcholine, so it cannot perform its function unless there is much more agonist than antagonist.
Some agonists of NACHRS are exogenous, such as choline and cytosine, as well as some antagonists, including chronotoxin and bungarotoxin. Thank you.